Okay, hi everybody, welcome. My name is Robert Froemke. I'm at NYU. I'll be moderating today's session with Andrea Gomez from UC Berkeley. So Andrea did her PhD at NYU in Steve Burden's lab, and then did a postdoc with Peter Schiffele in Switzerland. And now she's really just started her lab at Berkeley. And her work is exemplified by beautiful approaches in looking at the molecular mechanisms and signals that build synapses, which is the topic of her talk today. One fun thing is that Andrea did her PhD in the lab next door to mine when I got my lab started, and she came over to build a slice rig to do some physiology in brain slices to look at the function of LRP4 in the hippocampus. And so she took a rig that was basically in parts and put it all together. And years later, we're still using that rig. Anyway, take it away, Andrea.
Yeah, thanks, Rob. Good memories of that old rig. Yeah, I mean, just to kind of, reiterate, like, a lot of my work that I'm doing here at UC Berkeley is focused on the molecular mechanisms that pattern the flux of ionic current as it's transmitted across neural networks. And specifically, I'm interested in the designation of synaptic properties because these elements profoundly affect the propagation and transfer of network activity. Now, my group aims to discover the molecular coding principles of synaptic organization, and today I'll present a novel framework for how we think synaptic organization is achieved. And as the title of my talk suggests, our framework incorporates alternative RNA splicing, a powerful mechanism that can expand the coding power of the genome. And I'll also share how we test ideas within this framework in my lab using various strategies spanning molecular genetics, electrophysiology, and functional imaging. And toward the end of my talk, I'll take a step back and show some evidence of how synaptic activity interacts with and changes the intrinsic activity of a neuron. And here to demonstrate one of our approaches is an entire mouse brain imaged by 2-photon tomography. And as many of you may know, the structure that we genetically labeled here is indeed the hippocampus. Now, here is another view of the hippocampus, a brain structure that is essential for memory and spatial navigation. Now, when we think about network activity propagating through this circuit, and it does so in this general direction, we know the activity is not continuous. In fact, it is interrupted. As activity passes from neuron to neuron through the circuit, inputs summate, they trigger action potentials and repeat. And this is why I like synapses, or why I'm so interested in the properties of synapses, because they have a profound effect in shaping the way that neuronal activity is propagated across a network. But what are the cellular mechanisms for organizing these properties? Well, for one, we believe that a significant amount of the network organization is predetermined. And we think that unique transcriptional programs that are activated in distinct cell populations direct both the assembly as well as the maintenance of a network. Now, it is my main goal to decode what molecular programs are contained in these cell type transcriptional repertoires that contribute to synaptic organization and function, and specifically those that contribute to synaptic properties. Now, if we zoom in on a single CA1 pyramidal neuron in the hippocampus, we can appreciate that it is targeted by many different classes of neurons. And we can also appreciate that there are very different levels of highly stereotyped synaptic specificity. Now, cell type synaptic specification enables all of these inputs to appropriately identify and synapse onto its target with subcellular precision. Now, each of these connections also have different synaptic properties. For an example, one may have a specific neurotransmitter that mediates excitatory or inhibitory currents. Another may target special components to the presynaptic terminal that tunes the probability of synaptic vesicle fusion with the membrane, resulting in a facilitating or a depressing synapse. Another synapse may have special plasticity features such that when triggered by a strong or specific stimulus, can increase the number of neurotransmitters in the postsynaptic membrane, thus strengthening synaptic transmission. So how are these properties established? And we believe that this occurs through transsynaptic signaling molecules or transsynaptic linkers, each expressed by the presynaptic cell and the postsynaptic cell, that when bound, bring and cis together the components of the presynaptic release machinery, as well as signaling to organize neurotransmitter receptors at the postsynapse. Now, these modules, which we envision as senders and receivers, act to drive overlapping elements of synaptic organizations, such as bidirectional adhesion, it could be presynaptic vesicle recruitment, active zone assembly, or postsynaptic stabilization of neurotransmitter receptors. And we also envision that these transsynaptic signaling modules or sender and receivers can be mixed in combinatorial ways to generate the diversity of synaptic properties that we observe in neural assemblies. Now, in our model, the limiting factor for how many different types of synaptic properties that can be achieved in the nervous system is simply the number of available senders and receivers. Which brings me to alternative splicing. So just as a quick refresher on the central dogma, before mRNA can be read into protein, it needs to mature. And introns need to be cut out and exons spliced together. And what can happen during this splicing process, diagrammed here, is that alternative splicing choices can be made. And this happens through RNA motifs and transacting factors which either promote or suppress the usage of specific exons, which then go on to make distinct mRNA isoforms. Now, relating this back to the synapse, alternative splicing has the potential to vastly increase the synaptic proteome, thus increase the modularity of transsynaptic signaling modules, or rather these sender and receivers. And as a final but critical aspect of our model is that we hypothesize that there are splicing regulators that are selectively expressed in different cell types that drive unique splicing events that shape synaptic signaling. Now, back in 2016, when I was a postdoc in Peter Scheifele's lab together with a fantastic student, Lisa Tromuller, we found evidence to support this hypothesis. So let me introduce you to the RNA binding protein SLM2. And for the reasons stated before, we became extremely interested in SLM2, which is an RNA binding protein, because we found that SLM2 displays non-overlapping expression patterns in various cell types in the hippocampus. So we generated some SLM2 knockout animals, and we got motivated that perhaps SLM2 had some function in the CNS because they had a weird behavioral phenotype, in that they had an unusual response towards novelty. Normally, when a wild-type mouse is presented with a novel object, it will go and explore that object, here represented as an increase in time spent with the novel object. However, our SLM2 knockout mice displayed no interest in the novel object, and in fact, it actually preferred the familiar object, which is a perseverative behavior. And we didn't believe that this was caused by some type of like anxiety-like feature, like maybe the animal was like, didn't want to go to this novel object, it caused stress. Because when we tested them in different kind of anxiety, like our assays, in this one example, the elevated plus maze, showed that SLM2 knockout mice did not avoid the open arms of the maze. Actually, in fact, they actually kind of non-significantly but trended toward just staying into the open arms of the maze. So the loss of this RNA binding protein caused novelty defects in behavior. Are these changes associated with changes in transcriptome? And so to determine what, if any, changes that this RNA binding protein made, we performed a comprehensive global analysis by whole genome RNA-seq. And using Illumina paired end sequencing of libraries prepared either from wild type or SLM2 knockout hippocampus, we observed highly correlated expression levels of genes from knockout and wild type, indicating that the RNA binding protein does not tune overall RNA levels. Now, what about splicing? And here in this volcano plot where each dot is an exon, we can see statistical differences in excluded or included exons comparing wild type or knockout hippocampal tissues. And what we can, appreciate here is that only a handful of exons were deregulated. Now, at the time, this was an incredibly surprising result because most known RNA binding proteins regulate hundreds, if not thousands, of transcripts. And perhaps more impressively is that the deregulated exons encode for synaptic adhesion molecules, Neurexins 1, 2, and 3, and also the syntaxin binding protein Thomasin 2, all of which localize to the presynaptic terminal. Now the question is, do these isoforms have any function at the synapse? And to show what region or what exons were changing in the synapse, here is a qPCR of wild type or SLM2 knockout tissue. And what you can see that in wild type Neurexin 1, 2, and 3, if we take a look at this alternative splice site 4 diagrammed here, we can see that the isoforms are deregulated in SLM2 knockouts. I'm sorry, is it really pixelated on the screen?
It looks okay here.
Okay, sorry. This is really pixelated on my screen. I just want to make sure it's not on yours.
Just so, maybe a little blurry at the top, but I can make out the characters.
Okay, so if we take a look at which, where these exons that are deregulated, where they map to the Neurexin protein, we can see that these alternative splice site 4 position maps to a known ligand binding interface on Neurexin. So then we looked at, how this changed the function in the synapse. So when we looked at the components of the postsynapse and we observed a significant increase in the AMPA receptor subunit GLU-A1 measured on the surface of hippocampal tissue. Now, this coincided with an increase in excitatory current mediated by AMPA receptors. And when I subjected a whole cell recorded CA1 neuron to a special plasticity paradigm, which normally strengthens wild type synapses, SLM2 knockouts failed to undergo long-term potentiation, a process which critically depends on AMPA receptor regulation. Now, I set out to ask if alternative splicing was important for the specification of synaptic properties. And when we disrupted a splicing regulator by knocking out an RNA binding protein, we found that SLM2 is critical for a selective regulation at the RNA level, not upregulation or downregulation of specific genes, but specifically splicing was disrupted. Now, at the level of the synapse, we got a remarkably selective phenotype marked by a change in the level of AMPA receptors, which was coordinate with an increase in excitation. Now, since Neurexins were targeted by SLM2, we hypothesized that Neurexin splicing in the presynaptic neuron generates a transsynaptic signal that controls the magnitude of synaptic weight through AMPA receptors. Now, if this hypothesis is correct, then deleting SLM2 from CA3 neurons should produce the same phenotype. And taking this strategy is actually critical to rule out the possibility that the phenotype that we observe is coming indirectly from anywhere else in the circuit where SLM2 is expressed. And finally, to prove that the transsynaptic signal is indeed carried out by Nrxn, we went on to restore the wild type repertoires of Nrxn isoforms. And we would expect that this should restore the wild type synaptic function and behavior. So first, to probe presynaptic function, I made a conditional knockout by specifically deleting SLM2 from presynaptic CA3 neurons. And to achieve this, I utilized a cell type specific Cree driver that is selectively expressed in CA3 neurons in the hippocampus. So first, to test the selectivity of this Cree driver, cross this line into flocks TD tomato mice and assess the extent of Cree recombination using 2-photon tomography. And this is essentially the whole brain image that you saw on at the top of the talk. And this, you can appreciate just how selective this Cree driver is. Now, crossing this CA3 Cree driver into SLM2 flocks mice, we were able to generate a targeted deletion of SLM2 from CA3 neurons. And it is likely that this residual SLM2 positive neurons are interneurons that are spread out in CA3 region. Now, what happens to the splicing pattern in SLM2 deficient CA3 neurons? Now, before we did an RNA-seq and assess the RNA splicing pattern in whole hippocampal tissue. But we can't do that in this situation because we are only deleting SLM2 in CA3 neurons. And if we were to drop the whole hippocampus in sequence, we would get some contamination from other cell types where we're not manipulating the cell. So now to selectively look at the transcriptome only from CA3 neurons, we use the ribotag strategy, whereby ribosomes get selectively HA tagged in a Cree-dependent manner. We can then pull down ribosomes and assess the mRNAs. And if we look at wild type CA3 neurons here, this is a qPCR, and look at the Neurexin splicing, we can see that the splicing is almost entirely excluded at this fourth position. On the other hand, selectively deleting SLM2 from CA3 neurons results in a loss of alternative splicing at the fourth position. And now all of these Neurexin transcripts, Neurexin 1, 2, and 3, contain splicing at the fourth position, or contain this exon. Now, because of this highly selective expression of the Cree driver, we can directly test the effect of Neurexin splicing onto downstream targets because we think Neurexin is localized to the presynaptic terminal. Now, what I've shown you is, previously what changes to synaptic currents look like, but we also want to look at what other types of currents, like voltage-gated channels, things that like contribute to a spike, or the ones that determine the resting membrane potential, look like when we change properties of the presynaptic terminal. So now remember, I'm only deleting SLM2 from CA3 neurons. So the CA1 neurons that I'm recording from are wild type. And indeed, when I measure the magnitude of synaptic strength on wild type CA1 neurons that are contacted by mutant CA3 axons, indeed, I see an elevation in synaptic excitation, which confirms that splicing in the presynaptic neuron is instructing for postsynaptic strength. Now, as I mentioned before, what would we expect for the CA1 neuron to respond to this elevation of synaptic strength? And when recording from CA1 neurons, we can see that these wild type CA1 neurons that are contacted by SLM2 deficient axons, we see a decrease in the firing frequency in CA1 neurons. Moreover, when we look at other intrinsic properties like the HCN channel currents, or these IH currents, we see a significant elevation in these HCN channel mediated currents. However, intrinsic activity changes did not happen across the board. When we look at some of the active properties like the action potential, the dynamics did not seem to be changing. So it looks like even though we're seeing synaptic changes that lead to the decrease in firing frequency, the dynamics of these action potentials are not actually changing. And consistent with changes in IH currents and firing frequency, we also see a significant increase in the capacitance of these CA1 neurons that are contacted by mutant axons. We see a decrease in the resistance, and all this is consistent with what we would expect to see is a decrease in the time constant of the CA1 neurons. Now, to take kind of a broader look at what could potentially be changing kind of at a behavioral level, we put these animals through just the open field paradigm, and while most of the behavior didn't change as measured by the distance traveled in the open field maze, the number of crossings, we did see a significant decrease in the velocity of the animals navigating this arena, where only SLM2 was deleted in CA3 neurons. So to just kind of summarize what we saw with the intrinsic properties, which is that when we delete SLM2 from CA3 neurons, which we think targets neurexins, we see this elevation in synaptic excitatory synaptic strength. And coordinate with this increase in excitatory synaptic strength was increased IH currents, decreased firing frequency, faster time constants, and increased capacitance. Now, these results argue that specifically SLM2 in the presynaptic cell generates a transsynaptic signal that is responsible for a synaptic phenotype in the CA1 target pyramidal neurons. Further, that this transsynaptic signaling is also controlling the intrinsic properties of the postsynaptic neuron. Now, we believe that this is mediated by neurexins. But to confirm this, we would need to restore the correct Nrxn splicing in the presynaptic neuron. And to just remind you, the normal isoform contact in wild type CA1 neurons are Nrxn isoforms that skip this alternative splice for position. And when we delete SLM2 from CA neurons, we see this shift. So to restore the Nrxn isoforms, we would want to essentially try to make these isoforms that contain the exon and shift that back down to skipping. Now, to rescue this or to mimic this activity, we took a genetic approach. And here is just a diagram of the genetic approach. In using Nrxn 1 as an example, we have flux sites surrounding this alternative splice position. And anytime this position sees a Cree, it will essentially mimic the splicing activity as we would expect what SLM2 is doing. Now, to generate a rescue mice of all three Nrxins, I'm just kind of outlining the genetic strategy why we have a mouse that expressed the CA3 specific Cree with the SLM2 flops and that are wild type on this AS4 plus allele. And we cross them to SLM2 flox mice that are homozygous and flox allele at this AS4 position, with the goal of the rescue mice generating a mouse that has Cree in the CA3 neurons. We delete SLM2 entirely from these neurons, and we flox out, or we cut out one copy of the AS4 minus, essentially mimicking what we would expect in a wild type position. And indeed, whenever we restore the synaptic or mimic the isoforms that we would expect in a wild type neuron, whereas in the SLM2 knockout, we see an elevation in synaptic strength, as we saw before, restoring the Nrxn isoforms actually significantly drop the synaptic strength actually even further below wild type levels, and significantly diminish excitation of CA1 neurons, which confirms that splicing in the presynaptic neuron, again, is instructing for postsynaptic strength. Now, just to wrap up, as I mentioned in the beginning, I set out to identify molecular mechanisms that specify synaptic properties. And I share my view of how a synaptic adhesive code manifests in the hippocampus, and we kind of like to think about this as happening more broadly as manifesting in the central nervous system. But how robust is this code from mice to humans? How is the code manifested when you change the number of neurons by an order of magnitude from mice to humans? And this is why I'm interested in alternative splicing, because this mechanism can increase the coding power of the genome without disrupting the underlying evolutionarily conserved gene expression patterns and overlay another level of complexity. But for this to make sense for the brain, which is a highly heterogeneous system, increasing molecular complexity needs to happen in a selective way. And as I showed you, we discovered a dedicated alternative splicing program that devoted to the splicing of synaptic adhesion molecules. And when this was disrupted, not only did it alter behavior, but it also disrupted excitation and synaptic plasticity in the hippocampus. And importantly, by selectively disrupting splicing in a subset of neurons, we were able to demonstrate that alternative splicing can shape neural circuitry in cell type specific ways. And importantly, we had an RNA processing mechanism essentially control the postsynaptic properties of the connected cell. And this last point is especially exciting because then we can now go and dissect and molecularly profile what molecular mechanisms are responding to these increases in synaptic strength and result in decreased firing properties and elevations in IH currents. And finally, considering that 95% of multi-exon genes are alternatively spliced, this also points to the possibility that, there are other cell type specific mechanisms out there. And really, can different combinations of RNA binding proteins encode other aspects about synaptic function? Now, our discovery that alternative splicing can shape neural circuitry by selectively modifying synaptic properties is just one study in a growing body of evidence shown by us and others that alternative splicing is a powerful mechanism for network organization and function. And this is especially exciting considering that despite having a comparable number of genes amongst vertebrates, the extent of alternative splicing has expanded in species with more complex nervous systems, such as mammals. And actually the highest complexity is of alternative splicing, or the highest degree of alternative splicing is observed in primates, which positions alternative splicing as a potential driver for the emergence of complex networks. Now, from the scope of synaptic integration, small changes to synaptic properties like we observed in SLM2 mutants would have a big effect on the information flow to a neuron engaged in ongoing network activity. And then knowing this, it may not be so difficult to then imagine how circuits may be changed in neurological disorders where we know that synaptic dysfunction is disrupted. And this last point is especially relevant as disruptions to alternative splicing has been implicated in disorders ranging from autism to spinal muscular atrophy. And finally, understanding how splice codes are distributed across different cell types, we can then start to derive some cellular rules for how these splice codes affect neural integration and computation. And with that, I would like to acknowledge the lab members in my group, Adrian, Maura, and Anjali, and especially Peter Scheifele and Lisa Tromuller, who a lot of this work was done in the University of Basel in Switzerland, and also for my funding. And of course, you for your attention.
Thanks, Andrea. That's fantastic. It's remarkable the level of control you have over specific connections. So we have some questions. For one, what are you thinking about in terms of potentially IPing SLM2? Are you going to follow up with sequencing or mass spec?
Mm-hmm. So I guess maybe is the question to, I mean, the benefit or the idea of IPing SLM2 or what we could learn from that is, what is SLM2 interacting with potentially?
Right.
Now we know that SLM2 already targets Neurexins by direct binding assays, but we actually don't know, how is it that SLM2 biases the incorporation of, that AS4 region. And, potentially there are other factors that SLM2 is interacting with so that the RNA that exon is essentially being skipped. So yeah, I think that we could definitely learn a lot from understanding kind of the splice, or rather we would learn a lot by IPing SLM2 and understanding kind of what splicing factors are interacting in that engagement.
So you showed in the SLM2 knockout an impairment in LTP, which I think was induced by maybe theta burst, some kind of tetanization, right? Mm-hmm. What about other forms of synaptic plasticity? In particular, the question asks about spike timing or input timing dependent plasticity. Is it because the strengths are already kind of maxed out, or do you think there are other ways of modifying the synapse?
Yeah, so we haven't done any type of other plasticity paradigms yet, except for short-term plasticity, which we didn't see changing in our mutants. And yeah, the kind of current hypothesis is that because we saw this elevation in AMPA receptor at the synapse, we think that the excitation has essentially, or rather the plasticity has kind of reached a ceiling. And we hypothesize that AMPA receptors are not localizing or increasing at the synaptic membrane. But another plasticity that we are really interested in, what are essentially the next experiments that Andrea is going to do, are looking at input dependent plasticities. So if we consider the CA1 neuron as where it sits in the hippocampus, it also receives input from the entorhinal cortex. So we would expect that, because we're biasing all the synaptic excitation from CA3, that now the CA1 neuron is going to be is unlikely to kind of receive the entorhinal cortical input in kind of the normal timing fashion. So we would expect there to be a deficiency in the input dependent plasticity.
Yeah, so on that note, I mean, is that how you're thinking about the changes in the intrinsic properties? That they're compensating for one set of inputs changing, which might be a driver input, but that could impact either positively or negatively the integration and arrival of other inputs.
Yeah, I mean, at this point, we don't know if it's a direct or indirect kind of compensatory mechanism, whether or not the neuron kind of senses, that there's this increase in excitation, therefore it maybe, increases or decreases specific genes to make sure that firing frequency goes down and IH goes up, which, should essentially balance this elevated excitation. Or, if it's direct, maybe that there are some kind of local signaling pathways that are engaged that elevate these specific intrinsic electrical properties. But that's actually a project that Maura McDonough, a student in the lab, is going to do, is essentially molecularly profile CA1 neurons, these wild type CA1 neurons, and see not just what gene expression changes are happening, but also look at more in-depth kind of alternative splicing.
Yeah, for sure. So how complex is alternative splicing? How much kind of variability does this provide? Specifically, is it enough to express, to give all the synaptic varieties that you might see in the hippocampus and CA1, CA3 in the brain?
Yeah, I mean, I think the what we have in terms of the role of alternative splicing, or the role of diversity and complexity, is at this point correlative. What I think is kind of evident, or maybe some evidence, is if we consider just the Nrxn gene family, from 3 genes, 6 alternative splicing positions, and 3 promoters, we go from 3 genes to 12,000 different potential isoforms. And this would vastly expand the potential interactions that these Neurexins can make, and we're just focused on Neurexins. So I think it's certainly possible that if we look at other transsynaptic signaling systems, that we would see a similar trend, especially knowing that alternative splicing, or kind of evolutionarily conserved alternative splicing positions, target protein or ligand binding domains, post-translational modification, and intrinsically disordered domains, which are posed and positioned to really affect signaling mechanisms drastically. And that's just one aspect of alternative splicing. So we're not even talking about the non-coding aspects about alternative splicing. So we can get alternative 5' and 3' UTRs, which can dramatically affect RNA stability. It can affect RNA localization. So there's a lot of ways that alternative splicing can dramatically expand essentially the processing and signaling of these RNA molecules.
In the RNA-seq data from the SLM2 knockouts, you highlighted SLM1. It looked like there might have been a 50% increase or so. Do you think that's real? Is there some compensation at that level, or?
Yeah, so Lisa Tromula, the student that I worked with on this project before, published a paper in 2014 that showed that SLM2 negatively regulates SLM1. We know that SLM2 binds to SLM1 RNA and initiates a nonsense mediated decay pathway. So what that translates to is that anywhere there's SLM2, SLM1 is going to go down, and that's actually what we see in the splicing data. Is you see that that exon SLM1 go up because we have a diminished SLM2.
In the experiment you showed up towards the end, the rescue experiment, where the synaptic strength capped out lower than in wild types. Mm-hmm. I mean, the combinatorics are a little bit crazy, but have you tried to do subsets of the Neurexins, not rescuing 1, 2, and 3, but just one at a time? Do they kind of sum up linearly in terms of potential for rescue, or is there one that's more of a dominant isoform?
Yeah, so just by expression, Neurexin 1 is the most abundant isoform in CA3 neurons. And when I did single rescues, I saw a similar trend of it actually kind of overshooting and going all the way down. And now I haven't done kind of the combinatorial ones where with Neurexin 2, Neurexin 3, are kind of different combinations of those. I haven't tried yet, in part because these mouse genetic experiments are quite tedious. It took me just 3 years to generate this mouse, this rescue mouse. But I think that it's actually a very relevant question. We don't know how Neurexins are being distributed to all the axon terminals. We don't know if some Neurexins are targeting to CA1 neurons, or maybe some are targeting to PV neurons, or maybe some are not localized to terminals at all. Maybe there are some mechanisms that regulate whether or not a Neurexin is trafficked to a terminal. We have absolutely no idea about these things. There has been some studies, super resolution studies from Sudov's lab looking at Neurexin distribution and potentially showing that they're not at all synapses, which is actually really interesting. So for that particular question, be extremely relevant to know if they're affecting the function of the targets. And in that particular experiment, as you suggested, could potentially answer that.
Okay. Another question. Is there good pharmacology for Neurexin? Are there agents you can use to kind of regulate it in the moment?
Unfortunately, no, not that I'm aware. I know that there have been multiple attempts to generate antibodies to target these, but at the moment, no. I think some potential avenues that I'm particularly interested is the use of these CRISPR epi editors, essentially to target the regulatory regions on the DNA and essentially try to shut down or change the potential expression of Neurexin. But in terms of kind of acutely changing their patterns, it's been kind of difficult. And as you can imagine, there's so many isoforms. So which one do you choose?
Okay, thank you so much. With that, I think we'll close the session. Thanks, Andrea, for a fantastic talk. And thanks to all of you watching in Zoom and YouTube for the questions. And have a good rest of the day. Bye, everybody.
Bye.